Welcome to Introduction to Ecology. My name's Dr. Bewick, and I will be your instructor for this course. So a little bit of course information before we begin. My office is in 142 Long Hall. You can always contact me by email at sbewick@clemson.edu. Just a word of warning, I do not answer anything to a g.clemson.edu address, so please do not stick that G in there. You will not get responses, you'll be very upset, it'll be sad. So our meeting times, Tuesday and Thursday at 8 a.m. and I feel your pain. It's pretty early. Office hours are by appointment, but we will also have review sessions that are held right before the exam, a couple days before the exam, and that should help you to prepare for the exam and ask any final questions. There is a textbook. It is a required textbook. If you have not yet, please go and buy it from the library, from the bookstore. It's a good textbook. It's got a lot of nice pictures, and I think it does a good job of covering Introduction to Ecology. So what everybody cares about? Grades. There will be 3 exams and a final, and each will count 25% towards your grade. Also note, and this may be different from some of your other classes, exams are cumulative, particularly the final, but any of your exams may be cumulative. Those of you who are signed up for honors or a graduate course, there will be an extra project for you. There's some details in the syllabus. We will talk a little bit more about that later on. So what is ecology? The very basic definition of ecology is the scientific study of interactions between organisms and their environment. And so we're going to spend an entire semester looking at different plants, different animals, different fungi, all the different realms of life, and how they interact both with each other and with the abiotic surrounding world. Now, because this was a bit of a problem in the previous years, I'm going to tell you what Introduction to Ecology, that's this course, is not. This is not a bird course. Now, on occasion, we will study birds, and that's part of ecology, how birds interact with their environment, how birds interact with other birds, how birds interact with bird predators like cats, and how birds interact as predators, eating, for instance, insects. But if you think this is a bird course meaning that it's easy, you are wrong. This will not be an easy course. And it's not at any school. When I took Introduction to Ecology, it was one of the 2 hardest courses I took that semester. And in fact, of the 2 hardest, I think it was the harder. And the other course that sort of ranked comparable was Introduction to Organic Chemistry. So for those of you who have taken that course, you know that wasn't easy. This is not going to be easy either. Don't think it is. Also, Introduction to Ecology is not a freshman course. So there is the word introduction, but introduction does not mean, again, easy. Introduction means I'm going to provide you with the foundational principles of ecology, and some of those are tough. Some of those are difficult, and you will struggle to get your heads around it. So I don't want you to think of this as introduction in terms of a course that's an easy sort of baby step beginner summary of ecology. I hope you had something like that in high school. I want you to think about the fact that this is a 4,000 level course, and you should expect that the difficulty will be commensurate with that. This is sort of a bit of a warning as well. What Introduction to Ecology is, it's going to be a lot of math. You are going to have to solve basic algebraic equations. If you do not know how to do that, now would be a time to think about learning or possibly dropping the course. You're also going to have to learn to solve, or know already ideally, how to solve exponential and logarithmic equations. If these things scare you, again, get on it now, learn how to deal with these things now. You should have learned it in high school. Don't leave it until December when you're going to be really struggling. You also are going to need to know how to interpret graphs. If graphs terrify you, again, this is something you want to head off early on, really work hard on trying to understand that. A lot of people think that ecology is butterfly catching. And in fact, ecologists do catch a lot of butterflies. But the fact is, what they do with that data, what they do with those collected butterflies is very quantitative. And so a lot of ecologists are very good at mathematics and statistics. And you're going to be expected to be able to do that in this course. All right, So another misconception about ecology in general is that it's something that takes place in exotic locations. Something that takes place in the tropics, in Costa Rica, in Hawaii, in Australia. And it's true. Ecologists do like to go to those places. Especially in the tropics, there's a lot of diversity, and we'll talk about diversity gradients later in the course. But the fact is, we can study ecology anywhere. We can even study ecology in downtown cities. And I think one of the best examples is Rob Dunn's group at NC State, where they're actually looking at ants in Manhattan. So in Times Square, if you think about the place you would least expect to find ecology going on, it's probably Times Square. But the fact is, there's interesting ecology going on even there. So we can study ecology anywhere. And on that note, what we're going to do through this semester is we're going to participate in an ongoing project to catalog species diversity right where we live, to try and understand some of the ecology, or at the very least, sort of the components that are going to make up the ecology in Clemson. To do that, what I want you to do is go to iNaturalist. This is one of the best citizen science projects that builds a citizen science database. I want you to go there and I want you to set up an account. And once you've set up an account, you sign in, and then you can use this to log and help to identify all the different biological organisms that you see in the world around you. So if you can do everything through the interface in the computer. You can take pictures and upload them to your computer and then upload them through the computer to iNaturalist. But what's even easier is that you can actually download an app, the iNaturalist app, onto your phone. And you can do everything through your phone. And I know how much your generation loves phones, so this should really be appealing. All right, so again, download the app, open the app, and then anytime you see interesting fungus, an interesting plant, an interesting animal, whatever you can take a picture of, it's got to be bigger than a bacteria, unfortunately. Whatever you can take a picture of, take a picture, and you do that by just clicking on the camera button. All right, that's going to open up a typical phone camera. You're going to try and get the organism in the field of view, and then you're going to click on that green button. That will show you what your picture looked like. This looks kind of terrible, so you might want to retake it. But if you get a good picture, you'll click Next. And then you move on. Now this is a really cool feature that iNaturalist added very recently. You don't actually have to do a lot of digging through pictures online or through guidebooks. What iNaturalist will do is it'll take a look at your picture and it will use deep learning algorithms, again something very computer science-y, very quantitative, and it will actually try to identify what you found for you. And so you can go ahead and try and identify it yourself, that's great. But you know what most of you are probably going to do is click on the What Did You See? View suggestions. And when you do that, it tells you, it's pretty sure it's a particular thing. It might tell you at the tribe level, it might tell you at an order level, it might tell you at a class level. Depends on how convinced this deep learning program is about what you've taken a picture of. And then it'll give you the top 10 species suggestions. And so I looked at this and this happens to be a picture of a spider, and it's pretty close to this twin-playing jumping spider. And not only is this visually very similar, you can see the word visually similar, but other people have recorded it close by, which means it's probably in range. If you take a look at the genus down below, it's visually similar, but people haven't necessarily reported that from this area. So you can't always guess, and people on iNaturalist, it is a citizen science database, so there are citizens out there who are going to be looking at your pictures and saying, yes, I agree with that identification, or no, I don't. And some of them are going to be amateurs, total idiots, but a lot of them are actually professionals in that area, some spider people who study spiders for a living and go on there and look at your picture and say, yes, I think that's what that is. So you can click on that, and then what you should notice is that that appears in your list of observations. So I want you to record your observations, and there is a spreadsheet that you can download from Canvas that has sort of the format. You're going to record the date you observed it, what you ended up deciding the identification was. That can be based on you searching through the internet, or it could just be based on you picking one of the top 10 suggestions that iNaturalist already suggests. Now I will tell you, if you take terrible pictures, iNaturalist will not give you any decent suggestions. So you have to take a good picture. You got to put some effort into this, even if you don't have to identify it yourself. And then I want you to go back in and look if the community has actually changed your identification. And if it hasn't, just leave that. So I want you to fill that out, and I want you to email this to me at the end of the semester. And if you do that, I'm willing to give extra credit. And I'm going to tell you ahead, you're going to work for this extra credit. But this is going to be a possible way for you to boost your grades. And it's the only extra credit you're going to ever get in this course. So don't beg, don't grovel, don't tell me you'll write essays. This is what you have to do. And the fact is, the semester ends in December when there's not as much life out there. So if you want to get extra credit, I suggest starting now. So the deal is, I will add 1% to your grade for every 200 entries that you upload or that you take a picture and record on iNaturalist. I will count a maximum of 5% extra credit that I will just add on to your grade at the end of the semester. There's one caveat though. I don't want an entire 1,000 entries of plants or an entire 1,000 entries of spiders, if that's your thing. I want you to think about everything around there. So I will only accept 200 entries per phylum that count towards your extra credit. Feel free to upload thousands of pictures of birds, but you're not gonna get a 5% bonus for only birds. You have to have 200 max in each phylum for it to count. If you don't know whether things are in different phylums, you're gonna have to look it up. That's something you're gonna have to do. I suggest a convenient Wikipedia page where it lists the different phylums of life, and when in doubt, look it up. All right, so that's my little plug for iNaturalist. Again, that's citizen science. It's a citizen science database. And I want you to participate in citizen science, both so that you know how citizen science works, and it's something that more and more ecologists are relying on because we just can't be everywhere. There isn't the money for us to study everything everywhere, but interested and engaged people who aren't necessarily scientists themselves but want to learn about that particular realm can participate. All right, so to try and make you somewhat trained in ecology, let's move on to some important definitions that you'll need for the rest of this course. So basically we're going to divide the world into two. We're going to talk about the biotic component, and that means it's living or alive. So things like bacteria, they're small, but they're still living. Things like trees, it's not a charismatic bird flying around, but it's still a living thing, it's still biotic. Our favorite animal at this campus, tigers. And then we have the abiotic. And the abiotic matter because they impact what goes on with the biotic world. So things like temperature, things like precipitation, things like the minerals and chemicals and geology that make up the different soils throughout both South Carolina and the world in general. All of that is abiotic, and it matters to the biotic world, so it matters to ecology. And that's where we think about living organisms interacting with their environment. Their environment is often just abiotic, but it matters. All right, within living things, we can again divide them into two very broad classes. So all living things either fall into autotrophs or heterotrophs. Autotrophs are primary producers. They make their own food from energy that they obtain from an external non-living source. So the most common we think about is the sun, and all plants are autotrophs because most plants, I'll rephrase that, we'll learn a little bit more later, but most plants are autotrophs because they take in energy from the sun and then they convert that into their own food. There are also bacteria who take these abiotic, non-living sources of energy and turn them into their food, but it's not the sun. In fact, they use chemicals, chemical bonds, inorganic chemicals that are just in the ground, part of the soil. Those are also autotrophs. Heterotrophs are consumers. They do not produce their own food. Instead, they get that energy that they need. All organisms need energy. They get that by eating other animals. And they can eat other autotrophs or they can eat other heterotrophs. As long as they're eating something else, they're not making their own energy from an inorganic or an abiotic source, then they are heterotrophs. All right, some more important definitions. Thinking about energy and where organisms get their energy, we can divide energy into gross primary productivity, which is the total chemical energy captured by all the autotrophs in the world. So that is the autotrophs, again, are the organisms that take these abiotic sources of energy and turn it into food. So the gross primary productivity is all the energy that they capture. The net primary productivity is the net chemical energy produced by autotrophs. So autotrophs capture this energy from abiotic sources like the sun, but then they use some of it. And so that isn't available to other organisms. They use it to build their own tissue. They use it for their own metabolism. And so the difference between what they take in and what they use themselves, that's sort of the excess or net that's left over, and that's what we call the net primary productivity. Now, net primary productivity is very important because it sort of determines a lot of things like the biodiversity in the area, what things can grow in an area, and it actually changes throughout the season. And so here, if we take a look at this map, the darkness of the green is the degree of net primary productivity that's being generated in these regions in January. And what you'll notice is that we have a lot of net primary productivity in the equatorial regions where it's still warm and humid and lots is going on. If I think about where I live up in Canada, there's absolutely no net primary productivity in January. And that makes sense because the plants are all pretty much covered in snow. So there's not a lot of harvesting of sun energy up there. And so that determines what can happen in terms of the ecology in that region during the winter. If we move to March, what you'll notice is that that green net primary productivity shifts up. We see South Carolina starting to green up. We see some energy being captured in South Carolina by the autotrophs by the time we get to March. Again, by May, it's spread all the way up, even to Toronto. The snow's gone. We got plants sort of starting to come out, and they're starting to capture sunlight energy. And so what we see is net primary productivity goes up in May. We get to July, and what you'll notice is that it's really green in around the Great Lakes, the Toronto, Michigan, Maine kind of area. And so we got a lot going on there, a lot of energy being harvested. What happened to South Carolina? South Carolina has actually gone a little bit white there. That means we've dropped net primary productivity. We're not capturing as much, and that's partly because we have a lot of heat stress, and that makes the plants less able to capture the energy. They have to conserve water, so they have to think about, well, I can't photosynthesize now because if I did, then I would dry out and that would be the end of me. We get to September and we see that we're sort of, that net primary productivity in the northern and the high northern and low southern hemispheres is starting to decline again because it's getting colder. Some of those regions were actually starting to get snow again. By November, we see South Carolina still a little bit green. We've still got some energy being captured there. The equator, of course, is always green. And then we take us back to December and January. So what we see is that we have variation in net primary productivity spatially. So what's going on in Toronto is different than what's going on in South Carolina, even at the same time of the year. We also see that it varies temporally. So we've got really strong net primary productivity in the sort of southern Canada, northern US in July, but we have nothing there in December. If we think about South Carolina, we have, quite a bit in May, but then we don't have as much in July because it's too hot. So it varies temporally depending on your location. And then this impacts ecosystem function because that's sort of, the energy is what all the abiotic things need. And so the amount of energy that you have coming into the system at any time or any place is going to determine what can happen there to the biological organisms. So that's energy. And energy matters. Energy comes in, it's used up, it goes away. The other thing that's important are nutrients. Now, unlike energy, they don't come in and then disappear forever. They actually come in and stay and go around and around in what's called nutrient cycles. If you think about the sun's energy, it comes in, it's used up. That's the second law of thermodynamics. If I think about chemistry, if I have a carbon atom, it never really goes away. It's just always there. It might be changing form. So it might be part of a sugar molecule at one point, it might be part of a carbon dioxide molecule at another point. It goes around and around in these different forms, and eventually you close the loop and it becomes the same form again, and that's called a nutrient cycle. And so nutrient cycles are the cyclic movements of nutrients. The most important in ecological systems are nitrogen, carbon, and phosphorus. And they can cycle both within organisms. So, if I'm a carbon molecule in a person's skin tissue, or I could be part of the physical environment. I might be a carbon dioxide molecule in the atmosphere. And so nutrients are incorporated both into the biotic organisms and into the abiotic environment, and they cycle between them. All right. So that's sort of a broad ecosystem perspective. Now we're going to think about biological organisms and some of the processes that matter to them. And these are sort of fundamental processes to understand for understanding all of ecology. And the first is evolution. And there's a couple of different ways we can understand evolution. First, we can think about evolution as a change in the genetic composition of a population over time. So a classic example are moths in an area in England, I think it's Manchester. And so the moths started out white, and there was a couple of gray ones in there. And everything was happy until the Industrial Revolution, when we had a lot of pollution. And all that pollution made a lot of gray soot everywhere. And so all of a sudden, with the trees all covered in gray soot, the white moths stood out. And so what did birds do? Birds saw the white moths and they were like, let's go for them. And so they ate them. And what that did is it gave a positive advantage to the gray moths who blended more in with the sooty trees. And so what you saw is over time a change in the moth population, so that we went from having mostly white moths to mostly gray moths. There's some debate over whether that was the exact mechanism or whether that actually happened, but that's sort of an idea of how evolution works, even if this particular common example that people pull up may have had other factors at play. Another way that we can think about evolution is accumulation of differences from ancestors, and this is descent with modification. So my ancestor may have been a small little butterfly or small little moth with tiny wings and little antennae, but maybe that wasn't that advantageous. So, over time, the population changed, and so we ended up having a population where I had large forewings and antennas with big stars that helped to sense things. And so again, here we're not looking at change in the morphs or the types of individuals in a population. We're actually looking at change in the entire population over time as you accumulate, not just one change, not just going from white to gray, but a whole pile of changes. Maybe I've gone from small front wings to large front wings, antenna with no stars to antenna with stars, and forewings that are white to forewings that are gray. That's a whole pile of little changes that eventually make these things very different from one another. So that's evolution. And one way that evolution works is to sort of select on what are known as adaptations. And adaptations are characteristics of an organism that improve its ability to survive. So we talked about these moths being white and gray, and we talked about the gray moths blending in when we started to get soot accumulation on trees. So the color gray is then an adaptation that allows the moth to be camouflaged. And so that's what allows evolution to work. Evolution works on these adaptations and eventually selects for one adaptation over another. Natural selection is the process by which individuals with particular characteristics survive and reproduce at higher rates because of those characteristics, because of those adaptations. And so in this case, when we have predators picking off all the white moths, then the gray moths have adaptations or characteristics that allow them to survive, to not be picked off by predators. The fact they survive longer means they lay more eggs, and that means they breed more, they reproduce at higher rates, and eventually they take over the population and the white moths go away. So here we have a bird picking off the white moth, and if that happens over and over and over again, all we're left with is gray moth and gray moth babies. And so, if that happens for many, many years, we're left with a gray moth population. All right, so how is ecology organized? When I think about ecology, I can think about, a big scale, the ecology of the entire globe, or I can think about, the ecology of ants on a Manhattan street. So typically we sort of organize ecological processes in a hierarchy. And one of the first things after individuals that we talk about are populations. Populations are interacting individuals of a single species living in the same place. So here we have a population of mallard ducks. They're all the same species, and they're all living in one particular location. It's not mallard ducks in California and mallard ducks in Maine. It's mallard ducks on Lake Hartwell, or mallard ducks in South Carolina. That would be a population. A community is interacting populations of different species living in the same place. So if I think about a community of organisms that live in Lake Hartwell, maybe it's the mallard ducks, but we also have frogs, we also have lily pads, and we have flies. And so, the frogs are eating the lily pads, the frogs are eating the flies, the frogs are living on the lily pads, the ducks are munching on some lily pads, and so everything's interacting in that one location on Lake Hartwell, but it's a pile of different populations of different species. An ecosystem is a community of organisms, so all of those different species that are interacting with each other, plus the physical environment they live in. So now we're not just thinking about the biotic organisms, we're also thinking about the lake itself. We're thinking about the soil or the mud in the lake. We're thinking about the atmosphere and how much oxygen is there and how much nitrogen is there and how many pollutants and fine particulate matter is there. That's the ecosystem. We can then move to a landscape, which is one scale up again, which is a collection of areas that vary substantially and include multiple ecosystems. So we might have Lake Hartwell, but we could also include the experimental forest. And we could include, if we go even further, the sort of mountains in Asheville. We're all in a general area. They're all kind of within the state or just, across the border, but we have different types of environments. We have mountain areas, we have lake areas, we have swamp areas, all in the same broad geographic region. And then going all the way up to the scale of the globe, we have the biosphere, which is all living organisms on Earth, plus the environment in which they live. So that's how ecology is organized. And there's no such thing as an ecological rule, but there are a couple of principles in ecology that are pretty generally true. And so, we think of these as ecological maxims. They're sort of how we understand ecology as a process and sort of the predictability of ecological processes. The first is that you can never do just one thing. Remember on the last slide, we talked about communities and how communities are interacting populations of species. And that interaction word is a key and fundamental idea. It's like a web. It's like a spider web. If I push on one side, I get jiggles all through the web, even out to the other side. And so that's the idea with ecology. I can't just target one organism and hope that, nothing else changes. If I try to kill one organism, or if I try to boost the population of one organism, other organisms are going to feel a ripple effect. And so there's a classic example where we had caterpillars that ate the roofs off of villagers' houses. And so we had overpopulation of caterpillars, and that was it sort of seemed to come out of the blue, but in fact, the reason we had an overpopulation of caterpillars and the destruction of people's houses was because we had tinkered with the environment and tinkered with the ecosystem in other places, and that had had a trickle-down effect onto the caterpillars. Trickle-down effects may not work in economics, but they do in ecology. So the first thing that had happened is that we had gotten rid of the wasps that sort of attacked those caterpillars. And the reason we had done that, we had not purposely targeted these wasps. They weren't really doing a lot of harm, except to the caterpillars. But we had sprayed DDT to get rid of mosquitoes. And what did the DDT do? It killed the wasps. And once you killed the wasps, unfortunately the DDT did not kill the caterpillars so well, so we relieved pressure on the caterpillars, and the caterpillar population boomed. Now you might think, okay, well that was bad. But the fact is, adding DDT to kill the mosquitoes didn't even just affect caterpillars and wasps. It actually affected other species in this ecosystem as well. So the second thing that DDT did was that it accumulated in the geckos. And so what that did was, once you had all this DDT poison in the geckos, the cats that ate them died. And when the cats that ate them died, what did cats eat? Cats eat rodents, they eat rats. And so we had a huge rat outbreak, and then the rats spread plague. Don't forget, diseases, bacteria like bubonic plague are part of the ecosystem as well. And so, I'm tinkering with something as big as a mosquito, and I end up affecting not just other insects like caterpillars, but I end up affecting rats, which are mammals, and all the way down to plague, which is a bacteria. So again, you push on one place, and sometimes it's hard to predict, but you almost always know that something else is going to change. And that's something we need to think about when we think about conservation, when we think about land management, when we think about forestry, agriculture, all of those things. We have to think about, if I do one thing, what are these other effects I'm going to have? Can I predict them? And if I can't, what should I do? Is it worth the risk of interfering with that system, or could I be doing more harm than good? And there's a question of whether plague or malaria is worse. That's sort of a question that other people can answer. But the idea in ecology is that, affecting one thing changes another. Another ecological maxim is that everything goes somewhere. So other than energy, which disappears out of the system, everything else is kind of with us for life. And that means the life of the planet. And so, one of the worst examples of this is plastics and trash. And so we know that in the Pacific, there is a very, very large, what's known as a garbage patch. And if you think about Texas, which is the largest state in the US, this garbage patch is more than twice the size of Texas. And so when people throw out garbage, it doesn't just disappear, right? And so sometimes it ends up in our streams and into our oceans. And then all of that just floats there indefinitely. And that's what's caused this garbage patch. And so that's something we need to think about when we think about doing things to the environment. When we add these plastics and garbage to the ocean, that's there for a very long time. It's not going away. And that's affecting those ecosystems. And recently we've heard about microplastics in rain, microplastics in our drinking water. And so for a long time it's been damaging ecosystems. Now we're starting to realize it might be damaging ourselves. And again, that's that idea with ecology. We're all interconnected, and that includes humans. So when we do something to one part, sometimes it means it's going to have an effect, and often a negative effect on us. Okay, the third ecological maxim is that no population can increase in size forever. There's limits. At the very least, there's spatial limits. Things just don't fit anymore. But before you reach spatial limits, there's often resource and energy limits. So you might run out of energy. Humans worry about this a lot. Are we going to be able to grow enough food for ourselves? If we can't, we've basically run out of energy. Our population has grown so big that we can't supply the energy we need to keep growing. Before you run out of energy, sometime you'll run out of other nutrients. So for instance, plants growing in fields, they may have access to tons of energy. They got lots of sunlight. But if there's not enough nitrogen in the soil, they may be competing with each other for nitrogen, and that's often very limiting. That's why we add nitrogen as a fertilizer, because frequently these plants are, their growth, the number of plants you can grow in a field, the population size is limited by how much nitrogen is there because the plants need nitrogen to build their body. Common example is outbreaking insect pests. So things like the Douglas fir tussock moth, forest tent caterpillars, you may have seen these. You will get these large population explosions, and then the population runs out of food, or a predator takes over and starts attacking it, and they crash. They can't grow forever. In the early 70s, this became an idea that we sort of transferred from outbreaking pest species to humans, which you may think of as an outbreaking pest species. We are currently at a very high outbreaky kind of population, and we're doing a lot of damage to the places we live. And so in the 70s, Paul Ehrlich, who's a very famous ecologist, wrote a book that said we just can't keep growing. And the question is, what are we going to do? How are we going to deal with that problem that we're going to eventually run out of resources? Whether we run out of energy resources in terms of our ability to grow food, whether we run out of space resources in terms of places that we can live that are sort of sustainable and not in dangerous zones. All of these are questions that we need to deal with as the population grows. Another ecological maxim is that there is no free lunch. So energy and resources are finite. And if you increase allocation of a resource or energy to one function, then you have to decrease it at another. It's constrained. It's like your bank account. The more money that you spend on buying textbooks, for instance, the required textbook for this course, the less money you have to spend buying beer at the bar. And so some of you will decide to make a different monetary allocation than maybe you should, and you'll say, I'm not going to buy the required textbook because I can't buy everything, right? And I'm just going to take that money that I should have contributed to the textbook, and I'm going to allocate that to my beer fund. That's a decision you can make. I wouldn't suggest it. I'll probably test from the book. But again, that's that idea that you don't get anything for free. If you put energy or resources into one thing, like reproduction, then it decreases in another, like survival. Here's a classic example where this is actually humans, and they looked at life history trade-offs between the number of children you have, or your fertility, and the child survival. And so if you have, just 1 or 2 children, then you have a very high survival rate. And people that tend to have large numbers of children, 13, 14, at least in olden days, had a much lower survival rate. And so there's that idea that I can put all my energy and resources into having a few kids that I really focus on keeping alive, or I can put all my energy and resources into having a lot of kids and just hoping a couple of them make it. So that's the idea. The next ecological maxim is that evolution matters. It matters to everything. We talked about evolution in terms of gray and white moths, but it matters all the way down to small scale. And again, this is one of those places where ecology butts up on human health. So we are now talking a lot about antibiotic resistance. And the way that antibiotic resistance works is we have bacteria, and then we use antibiotics to kill them, but there's a couple of bacteria that have mechanisms to sort of deal with those antibiotics, whether they pump the antibiotics out at a really fast rate, whether they have a protective coat that doesn't let the antibiotics in the first place, or whether they just have sort of a change in the shape of the proteins that the antibiotic bound to so that those antibiotics can't bind and inhibit the cellular processes anymore. They have some trick that allows them to get away from the antibiotic. And so when you kill all the bacteria that are susceptible, you leave the fit organisms that were not susceptible to the antibiotic. And so what breeds? Those organisms that were able to tolerate the antibiotic. And so you get a bigger and bigger population of organisms that are drug resistant, and now you have no drugs that you can use. And so with bacteria, it's even more complicated because you don't just have evolution selecting on individual organisms. These bugs can actually pass their genes through a process called horizontal gene transfer. And that allows the antibiotic resistance to spread even faster. But the basic process is the same. We're selecting organisms that either have that or acquire it from their neighbor, have that ability to resist antibiotics, and then we end up with an entire population of antibiotic-resistant bugs. And hospitals are rife with these because antibiotics have been used so much in these places. Another ecological maxim is that time matters. So ecosystems change over time, and I think one of the most classic examples that's been studied by ecologists, including my previous advisor, is Mount St. Helens. So Mount St. Helens blew up in the early 80s, and it was one of the biggest volcano eruptions of the century, and certainly one of the biggest in North America. And so this is a picture of Mount St. Helens 3 years after the initial explosion. And what you can see is it's just dead. This is the blowout zone, and nothing survived. The trees are all dead, there's bare rock, there's no vegetation whatsoever. Go to 9 years, and if you squint, you can actually sort of see some greenery growing in there, in behind the trees. Very low, sort of grassy stuff in there. Now we go 15 years later, and we've got a dramatic change. We have the grassy stuff, it's sort of starting to take over. And then we actually even have some new trees starting up. And so you can see how much in 15 years Mount St. Helens changed. 24 years later, so almost a quarter of a century, and these trees are getting quite big. We're starting to get a lot more vegetation on the ground, on all the different sides of the mountain. 28 years, even more. We got some wildflowers growing in here, a lot of different diversity of new shrubs. And then, so basically, what you'll notice is that if I tried to study Mount St. Helens 3 years after the blast, I would have a very different picture of the organisms and populations living there than I do if I wait 30 years until after the blast. And so, this shows you that different places at different times are going to have different ecological processes, different organisms living there, same organisms living there even interacting in different ways. And so ecology changes even in the same place over time. And what that means is that past events affect the present and present events affect the future. Just like time matters, space also matters. And so this is a good example from pretty local. Mount Mitchell is the highest point in North Carolina. And you can actually take a hiking trail all the way up. You can also drive to the top. I learned that after I did the hiking trail. but it's a pretty steep climb. And at the bottom of the Mount Mitchell Trail, there's a sign, and it says, before going on this trip, even though it's only 6 miles or so, what you need to know is that it's a lot different in terms of the climate and the temperature up at the top of the hill than it is at the bottom. So if you feel hot and you think, I'm just going to need a t-shirt, that's good, I'll be good for the rest of the day, you might want to think about bringing a sweatshirt or a coat, because it could be really cold and rainy at the top, and you may not think you have a problem when you leave from the bottom. And so this is a case where the abiotic and biotic environmental conditions change dramatically from the bottom of the mountain to the top. And not only, again, it's much colder at the top of Mount Mitchell than it is at the bottom of the hiking trail, but because of that, the entire forest changes. We have a lot of deciduous trees at the bottom, sort of what you might see around this area. By the time we get up to the top, we're looking at spruce hemlock forests, much more conifers, and without as many deciduous, in fact, without hardly any deciduous at all. And so your biotic organisms respond to these environmental changes over space. And again, that means that your populations are different in the different places, your communities are different in the different places, and your ecosystems are different in the different places. Yes, and there we go. We enter stands of spruce and fir and a lot of conifers. So basically, it smells like Christmas when you get up there, which is not true of this area. All right. The 8th ecological maxim is that life requires species interactions. And if we just think about this from our own perspective, we are organisms living in this world. We interact with other populations. We are part of ecological communities and ecological ecosystems. We're part of that. And so we rely on plants, for instance, trees, to build our housing structures that we live in. We rely on animals and plants to provide us with energy resources in the form of food. So, we might eat certain animals like pigs around here. That seems pretty popular. Barbecue, it's a southern thing. Not so much in Canada, although we do eat a lot of meat. And then another very popular southern fare is peaches. And so again, we rely on these species. Sometimes we don't think of them as, ecological species, because we're cultivating them and we're growing them in agricultural systems, but it's the same thing. This is just a different ecosystem when we're interacting with these species. All right, so in ecology, we have these different processes happening at different scales, and we have these truisms that, apply to all ecological systems. But when we think about ecological processes and these truisms and what we can say about them, we have to think about the importance of scales. And so that means, are we looking at a small scale or are we looking at a large scale? At what level are we asking questions about a particular ecological process or a particular ecological phenomenon or even a particular ecological maxim? And so if you take a look at the area occupied by monarchs at overwintering sites, and you take a look at that over a, short time period, say from 2012 to 2017, you might say, hey, look at that. The monarch population is actually doing better. We're getting better. So that's a very short time scale. However, if your bigger question is, are monarchs on their way to extinction? Looking at 5 years of data is probably not the right scale, right? Because bad things have been happening for a lot more than 5 years. And so you need to think, well, maybe I need to look at a longer term trend to answer the question of whether monarch populations are plummeting to extinction or whether they're actually doing, okay, whether they're surviving or even getting better. So if I go all the way back to the 1990s, probably when you guys were, actually before you guys were born, what you'll see is that now the area occupied by monarchs at overwintering sites, now you can see a clear and dramatic and very terrible decrease in the amount of area occupied by monarchs. So again, if you look at a short time scale, it might make you convinced that monarchs are doing fine. If you look at a long time scale, not so much. So you want to select a time scale that's long enough to capture the trends that you're interested in. And sometimes that's tricky in ecology because again, if you folks were doing the experiment, you weren't alive in the 90s. So you think about your whole lifespan, it's tough to do an ecological experiment for your whole lifespan, but sometimes you need to get the right answers. Same thing with scale. So if we look at scale, we might look at where the butterflies are coming, and we might say, oh, they're all sort of coming from the center of the country. But then if we sort of expand out, what you'll notice is that there are butterflies coming from other areas. They're coming from the northeast, they're coming from the northwest. And so spatial scale matters if you're interested in, where are all these butterflies coming from. If you're only tracking them going up the Midwest corridor, you might not be understanding the processes that are leading to decline in the northeast. You may not be understanding the processes that are leading to decline in the northwest. Maybe the mid-central population is doing just great, and in order to understand the declines that are happening in the overwintering ground, we need to know what's going on in New York, or we need to know what's going on, in Alberta, that kind of question. All right. So the scale matters in terms of asking questions and in terms of designing an experiment. But even when you pick the right scale, you have to think carefully about how you're going to ask that question and what experiment you're going to do in order to ask that question in an accurate way that's going to get you accurate answers. And so frequently what ecologists will do is what's known as a controlled experiment. And what they do is they alter one or several factors and observe the effect. And so typically what they want is a control group where they don't do anything. That's sort of, you don't add fertilizer, or you don't add nitrogen or something like that. And then they have one or more experimental group that they actually do something to. They add fertilizer or nitrogen. Maybe they, change the amount of carbon dioxide in the atmosphere, something like that. And so to do that, you need to divide your whole area, your whole spatial scale of your experiment up into plots. And so you might put fertilizer in one, and that's your control plot. And then you might put fertilizer in one, and that's your experimental plot. And you might not put fertilizer in the other, and that's your control plot. And then you might see a whole pile of plants growing in the fertilizer plot and only one growing in the control plot. That's great, but there's a problem. Maybe there's something else about that side of the field. Maybe it just got a lot more sun, right? Because it got the sun coming up in the morning, and that's why, and it had nothing to do with fertilizer. And so this is not a very good experimental design because it doesn't have replication. Replication means that each control and each treatment is performed more than once. So it can't just be, well, there was something else that I didn't know about affecting that one plot versus the control plot, and that's why I saw the effect. So here I've divided my plots up, and I've got, a whole pile of plots, and maybe I use fertilizer on some of those plots, and I don't use fertilizers on the other. So I have my experimental plots in orange and my control plots in green, and again, I see tons of plants in the experimental plots and only one in the control plot. But if you look at that, you might say, well, but that's not really different from the last picture. You just kind of drew some lines, right? So you have replication, but that whole side may still be subject to more sun because the sun comes up on the east. So this is, again, a bad design. It's got replication, so we're one step better. But what it doesn't have is randomization. You don't want all your control plots in one place and all your experimental plots in another. Because again, whatever you're doing to manipulate that experimental plot, there might be something else going on if all of your experimental plots are clumped together. And so here we have a completely randomized experiment where we have orange plots with fertilizer, green plots that are control, and now we can't just say, well, it's because the sun comes up in the east. So if we see a lot more plants growing in the orange plots, then we can much more convincingly argue that fertilizer causes more plant growth. And so when you design experiments, you got to think about spatial scale, you got to think about temporal scale, and you also have to think about the overall design, and you need to think about replication. Well, first of all, you need to think about having a control group and an experimental group. Never, never, never do an experiment without a control group. You also still need to think about replication. So don't just have one control, don't just have one experimental, you got to do replicates. And then you need to think about randomization. You don't want some feature to be the same in all of your control plots that is then different in all of your replicate plots, or all of your experimental plots. All right, so that's the ideal world. We would love to always do controlled experiments. Remember I talked about spatial scale and I talked about temporal scale. And I said, to understand monarch declines, we need to have been studying monarchs for longer than your lifetime. And to understand monarch migration, we need to have been studying monarchs over the entire country. So that's rough. That costs a lot of money. Maybe we would have been able to do it if we thought about doing it in the 90s. But to have controls and replicates over large geographic regions and long time periods is difficult. And when we start thinking about questions at the global scale, remember that final scale of ecological research, the biosphere, when we start thinking about that, we can't do a replicate experiment. We have one Earth. And so a good example of experiments we'd love to have replicates of are climate change. Because climate change is a global phenomenon. We can't really look at climate change in one area because it's happening to the whole world all at once. I would love to tell you there are 6 Earths out there that we're monitoring, and we are adding carbon dioxide to one of them, or to 3 of them, and we are not adding carbon dioxide to the other 3. And then we would be able to know for absolute certain whether that carbon dioxide is leading to climate change and whether, those unperturbed globes are doing just fine. But we don't. There's one Earth. We cannot do replicate studies on the planet. So what do? In this case, you have to look at observational studies. And so we take a look at observations, and this is something known as phenology, or the timing of when species live their life cycles. And so we can look at a whole pile of different species. We don't have, multiple globes, but we can take all sorts of different amphibians and all sorts of different birds and all sorts of different butterflies and all sorts of different plants. And we can ask whether their timing is changing. And what we see is that 99% of them are moving to earlier dates. And that's in keeping with our understanding of what global change is doing. We're warming up the world. And so things that used to have to wait until it got, warm in May can now come out in April because it's warmer. And so again, we have many observations across many species that are consistent with our hypothesis that global change is altering species phenology, altering the timing of when species do their life cycles, their thing, and that alters how they interact with each other. So observations are important. Remember I said, these observations that we see, everything's coming out earlier, that's consistent with our understanding of what global change should do. Part of the reason we think that's what global change could do is because we have quantitative models, mathematical models. This is where math is important for ecologists, that predict that when I warm up monarch butterflies, or when I warm up moths, or when I warm up any insect, quite frankly, it almost always comes out earlier. It emerges faster, it lives a shorter lifespan. And so you can build things called degree days, and they are made by sort of studying individual life stages of insects in the lab and heating them up and seeing how long they take. You can put those together and build a model that predicts what's going to happen at a population or a community or an ecosystem level when you start warming things up. And so these models are mathematical hypotheses, basically. But when you see observations that are in line with these mathematical models, it gives you a sense, even though you only have one replicate Earth, it gives you a sense that maybe I'm right. Maybe my health, in fact, at this point, 99.9% of scientists agree climate change is happening. It's messing up the ecological world. And the reason they believe that is because they see observation after observation after observation that is in line with how they expect climate change to alter the world. And part of the reason they think that the world should be altered in particular ways is because they have the models to back it up. The models that predict, hey, this is what we think is going to happen, and this is what we see is going to happen. We see it happening, our models predict it. It's all these pieces of evidence that contribute towards that general idea that climate change is happening and it's messing things up. So this brings us to sort of a summary of the scientific method. What you do to ask scientific questions. You've probably seen this before, right? So you observe and you ask a question. Then you use knowledge or intuition to develop a hypothesis, right? I observe many organisms are shifting to earlier dates in terms of when you see them in the world. And so I think that this is because of climate change. That's my hypothesis. Then you perform experiments, you collect observations, and you build models, and you determine whether these all back up your hypothesis or whether you actually find out, hey, my hypothesis was wrong. And then you use your results to modify your hypothesis. Maybe your hypothesis was perfect. Sometimes it wasn't. Sometimes you got the big picture. Climate change is happening. But in fact, the biggest concern isn't that species are moving to earlier dates. The biggest concern is that when they do that, some species move faster than others, and then they don't interact anymore, and that drives them extinct, right? That changes your hypothesis slightly, it changes the focus, and that allows you to pose new questions. Well, hey, how hot does it have to get before these two species can no longer interact at all, before I don't have any predation on these caterpillars, and then their population is just going to blow up? So your book goes through a case study, and I think it'd be good if you read that case study. Again, it's a required textbook. Read it. And it doesn't talk about climate change, but it talks about an equally problematic thing we've been doing to our world. We've been, adding garbage and pollutants and all sorts of stuff. And so what happened, there were some students that noticed deformed tree frogs and salamanders. These organisms had extra legs, they were missing arms, it was terrible. So that's an observation, right? That's the first part of the scientific method. Simultaneously, some scientists noticed deformed leopard frogs. Again, extra legs, all sorts of, deformities. Some of the legs weren't shaped properly. Again, another observation. And then what they noticed, at least the scientists, the students didn't do this as much, is that all of these deformed amphibians tended to have Ribiera, which is a parasite. So that's another observation. This led them to the hypothesis that the Ribiera was actually causing the deformities they were observing. And so what they did is they took little glass beads that sort of mimic what the Ribiera does in the developmental process, and they put those into frogs. And so that was an experiment. And their hypothesis was that there was a second hypothesis. So that was one hypothesis, that it was this parasite. The second hypothesis was that the deformities were due to pollutants in the ponds. And so that's, again, an observation. What they found is that the ponds with the highest level of deformities actually had the highest levels of pollutants. So the final hypothesis was that the deformities were only in ponds with Helisoma, and that is a host for the Ribiera. So again, these are all observations. This led to the hypothesis that tadpoles exposed to more Ribiera would be more deformed. And so they did a controlled experiment on that, and they actually changed the amount of, they exposed some of their tadpoles to Ribiera, they exposed some of them to different amounts of Ribiera, and some of them to no Ribiera, and they found that, they did see deformities, but it wasn't sort of the extent that they expected. And so then what they did is they took mesh cages in the ponds with and without pollutants and showed that there was a synergy between the parasite being present. So the parasite did cause deformities, but it didn't do it quite so much unless the amphibians were also in polluted ponds. And so, again, this was another controlled experiment where they, put Ribiera in some tadpoles. They didn't put Ribiera in other tadpoles. They put some of the Ribiera-infected tadpoles in polluted ponds. They put some of the Ribiera-infected tadpoles in not polluted ponds. They put some of the control uninfected tadpoles in not polluted ponds, and they put some of the control uninfected tadpoles in polluted ponds. And what they found is that the tadpoles with Ribiera in polluted ponds were absolutely the worst. And so they came up with this idea that the pollutants suppress the frog immune response, and that allows the Ribiera to really take hold and cause the deformities. And so again, they did a controlled experiment. And so this is the scientific process. There were some observations they made. It led them to a couple of hypotheses. They did some experiments that were not controlled. Then it led them to some controlled experiments. And then they finally came up with this idea that a very complex system, it's not just a parasite that caused the deformities. It's not just pollutants that cause the deformities. Both of those did cause some level of deformity, but when you put the two of them together, you got this much stronger occurrence of deformities in the ponds. All right, so read through that example. It's a nice example of the scientific process looking at ecological systems where you have parasites and snails and amphibians all interacting with their environment, with the abiotic environment in the pond, with the pollutants that are present. And it's sort of a nice summary of the sort of ecological method and how that's applied to ecology. All right, so if you have any questions, email me. Remember, email at theclemson.edu, not the g.clemson.edu. I will not get your emails to the g.clemson.edu. email me with any questions. If you want to talk about ecology, if you want to talk about anything like that, I would be happy to take your questions there or stick around after class.